Microinjection. Microinjection is a technique that uses a glass micropipette to inject liquid substances into living cells or intercellular spaces. This process typically involves an inverted microscope with about 200x magnification, though other types of microscopes can also be used. For cellular or pronuclear injections, the target cell is positioned under the microscope, and two micromanipulators, one holding the pipette and another a microcapillary needle, penetrate the cell membrane or nuclear envelope. This method is commonly used for introducing vectors into cells, creating transgenic organisms, cloning, studying cell biology and viruses, and treating male subfertility through intracytoplasmic sperm injection. It allows researchers to directly inject genetic material into the nucleus of a target cell, making it a key technique for gene transfer in animals such as mice, rats, frogs, and larger organisms. Overview of the microinjection process. Preparation of genetic material. Prepare the genetic material, including the transgene and regulatory elements. Preparation of host cells. Collect and prepare fertilized eggs or embryos for injection. Microinjection. Use a fine glass micropipette to inject the genetic material into the cell nucleus. Control the micropipette with a micromanipulator under a microscope. Integration of genetic material. The injected genetic material integrates into the host cell's genome, often randomly. Culturing and implantation. Culture the injected cells and, if necessary, implant them into surrogate mothers. Screening and analysis. Screen the resulting organisms to identify those with the desired genetic modification using techniques like PCR and Southern blotting. Microinjection systems. Continuous flow microinjection. Delivers a constant stream of genetic material by applying continuous pressure to the injection pipette. Pulsed flow microinjection. Delivers genetic material in pulses by modulating the pressure on the micropipette. Pressure-based systems. Uses continuous or pulsatile pressure mechanisms to control the injection process. Electrode-based systems. Uses an electric field to facilitate genetic material entry, known as electroporation. Automated microinjection systems. Features robotic components for precise and efficient automated injection. Steps in microinjection. Micropipette preparation. Heat and stretch glass to form a fine tip, 0.5 mm in diameter. Microscope setup. Perform the injection process under a powerful microscope for precision. Cell placement. Place the target cells in a container. Cell holding. Use a holding pipette with gentle suction to secure the cell. Micropipette positioning. Mount the micropipette on a micromanipulator for precise movement and position it near the cell. Injection. Insert the micropipette into the cell membrane, cytoplasm or nucleus, and inject the contents by applying hydrostatic pressure with a microinjector. Withdrawal. Carefully withdraw the micropipette to minimize cell damage and ensure cell viability. Advantages of microinjection. No need for selection markers. Unlike other gene transfer methods, microinjection doesn't require antibiotic resistance genes, simplifying the process. Precision allows exact control over the volume and timing of material delivery, which is difficult with other methods. Easy identification. Injected cells can be marked with dye or fluorescently labeled proteins for easy identification. Reduced protein preparation. Requires less protein preparation than electroporation, beneficial for experiments with scarce or expensive proteins. Less cell stress, reduces cell death compared to chemical transfection or viral infection methods. Disadvantages of microinjection. Need for vehicle controls, requires vehicle controls to assess potential effects on cell viability and ensure accurate results. Technical expertise required, requires high skill to master and maintain cell viability. Alternative techniques popular, methods like transfection, infection, and electroporation are often preferred for delivering non-permeable materials into cells. Labor-intensive, manual microinjection is time-consuming and impractical for large-scale or high-throughput experiments. Limited scalability, typically involves injecting a small population of cells, limiting scalability. Inappropriate for large-scale genetic material transfer, not suitable for transferring genetic material into many cells for techniques like Western blotting or purification. Challenges with certain proteins, difficult to directly inject certain proteins, like membrane proteins or neurotransmitter receptors. Thank you.